Evening, everyone. Lovely to see so many familiar faces and some new ones too, I think. Another brilliant turnout. So we're really excited to be taking you through atopic dermatitis today. Those of you who haven't seen us before, my name's Josh. I'm the regional manager for AP. And today delivering the wonderful webinar will be Rebecca Edwards, who is our Director of Education, and Karen Harrison, who is our European Education Manager. So between them, they will take you through the webinar and we'll be around at the end for a Q&A. Those of you that missed it, Rebecca was just saying, we will be recording this. So as many of you as possible that can stick to the end, that will be fantastic. But if you do need to rush off, you will be sent a recording in a couple of days. Any questions, please? If you can use the chat bar, we will tabulate those and run through them at the end. And I will hand you over to the ladies without further ado. Thank you very much.
Thanks, Josh. And well, I would say good morning, everyone, because it is 5 minutes past 4 in the morning for me in Melbourne. I know. Thank you for those sympathetic faces. But do you know what? It's actually so exciting to be here. And those of you who are on the webinar I did last week at the same time slot, my feelings stand. There is no other bunch of people I would willingly get out of bed at 3 o'clock in the morning to come and talk to. And I've actually been looking forward to this so much that even though I was trying to put myself to bed early last night, I was so excited about talking to you all that I couldn't sleep anyway. So, it's no hardship for me to be up here in the middle of the night chatting away to some lovely, lovely people about one of my favorite topics, which is gut health and how those of us in the naturopathic world can harness its powers to help our patients. So I'm going to share my screen if Karen's given me the power. Oh, she has. Excellent. Okay, and let's get this happening. And yeah, just to repeat what Josh and Karen were saying, this is being recorded, so you'll all receive a copy if you need to run away or if you want to take, further notes in the future. And if you have any questions, type them in the chat box as soon as they pop up in your mind, and we'll answer them at the end. So what we're talking about today is the interrelationship between the microbiome and atopic dermatitis. And I think, one of the reasons we've got so many of you on this call and registered to receive the recording later is because atopic dermatitis or eczema is one of the most common presentations that naturopathic practitioners see in clinic. I definitely remember when I was in student clinic, this is what, this was kind of our bread and butter. When the clinic supervisors knew that an eczema case was coming in, they would say, right, we'll give this to the, the junior clinical students because this is going to be something you're going to need to approach for the rest of your career. And also, if you can effectively help patients with eczema or other allergic presentations, you'll always be in business because, well, I'll show you some stats in a second. The number of people, especially children, who experience atopic dermatitis and other aspects of atopic disposition, there are plenty of people out there who need our help. So as we at Activated Probiotics are a microbiome specialist company, this is an area where we immediately wanted to dive straight in and find out, right, what is the latest research? What can we do to help clinicians help their patients? For those of you who haven't met us before at Activated Probiotics, very briefly, we are an Australian practitioner-only precision probiotic specialist company. And that means that we use the latest evidence to create probiotic products which are focused on specific conditions. So we have a look at what's the research on coming out of all the different microbiome specialist areas in the world on specific strains of probiotics. What have they been found to do in the body, and how can we, like I said, harness that power to help clinicians help their patients? So today we are going to be talking about atopic dermatitis or eczema. Let's just go through the name first of all. Eczema is a word that we use, kind of pretty conversationally, but technically we really should be calling it atopic dermatitis. Dermatitis obviously meaning skin inflammation, and atopic referring to atopy. And this is acknowledging allergy, essentially. So it's long been acknowledged that eczema is part of an allergic presentation. It's not an allergy in the same way as, a specific food allergy, but it is part of a wider allergic presentation. What's interesting is, like I alluded to, this is a condition we really need to be comfortable with because it affects such a significant percentage of the population. Just in the last 30 years, which, looking at all these lovely youthful faces on the screen would be probably about the average lifetime of most of you. Just in the course of your life, eczema prevalence has increased 2 or 3 times. And this, it's perpetually being questioned by scientific researchers. Why? Why is eczema becoming more common? Is it just that more people are presenting to their doctors? Is it that we are better at, diagnosing or, collating statistics on it? Or is it actually increasing? And I think we're pretty clear that it is actually increasing. There's not one single reason why. And today, one of the things we're going to talk about is all of the different factors that driving the presentation of atopic dermatitis. It's definitely not a single factor condition, but one of the factors that drives atopic dermatitis and that is essentially very, very relevant to us is microbiome dysregulation. We've long made a connection between not just atopic dermatitis, but many other allergic conditions and what we call the hygiene hypothesis, which I know you're all familiar with. A little bit more coming on that shortly, but essentially increasingly dysbiotic environments play a big role in driving this rise in prevalence of atopic dermatitis. Somewhere up to around about a quarter of all children are affected by eczema. So, those of you who have children, I'm very, very certain that eczema has featured in your parenting life, either in one of your own children or, more than one, or in your parenting circle. It's very, very, very common, to the point where its frequency is pretty, I feel, almost doing a disservice to the severity of some sufferers. If you have a child or know someone who really suffers with eczema, who's repeatedly having skin breakdown, infections, itching affecting sleep, and the whole family's mental health, for someone to be like, oh, eczema, oh yeah, my kid gets that, that's nothing, can feel very dismissive. So it's always really important, I feel, when we have patients come to us whose life is being significantly affected by atopic dermatitis, that we just sit with them and acknowledge the severity of what they're going through. It's a real spectrum of severity. And while the majority of children will be towards the less severe spectrum, I just always want to acknowledge that there are people whose lives are really significantly impacted. And it's so wonderful when we can have a beautiful outcome for those people. And that's what I want to, deliver the information for you to build into your clinical practice to help those people. What's interesting is that while around 20% of children are affected, only about 5% of adults experience eczema. And that's one of the things we're going to have a look at today. Why is that? What changes? Really interesting. So as I just alluded to, eczema or atopic dermatitis can have significant negative impacts on someone's life. It's not always just a case of a bit of dry and itchy skin. I will always remember seeing cases of eczema in my clinical practice so severe. I remember this one woman who, she held up her hand like this, and it was like she was wearing like an overlay of wax on her hand, that she had suffered with eczema for so many years, that she'd built up all of this extra, essentially scar tissue. She had deep cracks between her fingers, and she was just constantly in this cycle of antibiotics and steroids, because as soon as she would clear one infection, another infection would open up between another pair of fingers, and she'd have these huge staph lesions taking over her arm. And that's, she couldn't sleep because she was in so much pain. She couldn't use her hands properly. That's not just a little itchy rash. And I'm really happy to say that we saw such significant improvements by looking at gut health with her. As with so many things in the naturopathic world, it always comes down to gut, start with the gut. So it can be very, very severe, although for the majority of people it is mild, but quality of life can be significantly affected. And something that's really interesting is that this actually has been examined in the research. Some of the studies done on one of the probiotic strains we're going to talk about today has actually specifically been looking at quality of life. and measuring quality of life in eczema sufferers and how that changes when they take probiotics compared to placebo. So that's something interesting. From a parent perspective, I'm very lucky that my child never had eczema, but I certainly remember in my NCT group, some of the babies in our group who did have eczema and the effect it had on their sleep and therefore their family sleep and therefore everyone's mental and emotional well-being. It's not something to just be easily dismissed. So I'm so excited to talk about this research and think about all the people who collectively we can all help. Right. So as we kind of mentioned earlier, there are lots and lots of different factors that feed into the presentation of atopic dermatitis. Sorry, I need to keep the tea flowing because I haven't had any sleep. Throat's a bit scratchy. So we've got at the top of this kind of wheel, what kicks off the moving wheel of atopic dermatitis is genetics. There is a really strong genetic element, but as with everything, what we've learned is genetics is not the only factor. It is never as simple as, well, your dad had eczema, so you're going to have eczema. It's never that simple. Yes, different genetic polymorphisms increase the risk and can change the presentation of some of the proteins in the skin, but it's not ever the only factor. We're talking about a really complex interplay between genetics and the environment. Now, a little bit more coming on that. Like I said before, the hygiene hypothesis plays a huge role here. And this is, oh my goodness, it's an area I could talk about for hours, the diminishing human microbiome and its effect on immunomodulation. So we are essentially living in an increasingly dysbiotic world, and it is no coincidence that the highest rates of atopic conditions correlate to higher socioeconomic status in first world nations or developed nations. Essentially, as we become more clean, our immune systems become less able to manage the pathogens and pathobionts which are presented to them, and immunodysregulation occurs. And what this can look like in atopic dermatitis is epidermal barrier dysfunction. Again, I've got some pretty cool pictures coming up on this. Nothing gross, by the way, it's all theoretical. Pictures talking about how the cycle of immune dysregulation and epidermal barrier dysfunction leads each to exacerbate the other. So once again, we see eczema patients becoming caught in this vicious cycle of inflammation, dysregulation, skin breakdown, each one making the other worse, and it can be very hard to break that cycle. The immune dysregulation that we're talking about, like I said, goes hand in hand with epidermal barrier dysfunction. We're very much looking at a Th dominant phenotype here. So essentially what we see is patients becoming stuck in a cycle of dysfunction between those T regulatory cells and Th2 to Th1 deviation. They're stuck not being able to deviate immature T cells or naive T cells towards Th1, and we see a very Th2 dominant presentation, which further enhances inflammation. That's the next point in our wheel. Further enhances inflammation, and when we have a Th2 dominant situation, this further promotes the breakdown of epidermal barrier dysfunction, and round and round and round we go. So, don't worry, we're going to talk about each of these in more detail, but just setting the scene for a really complex interplay of lots of different factors, each makes the next one worse. So a little bit more on the hygiene hypothesis. And I know this is a topic dear to the heart of many of us. And, I certainly remember when I had my little baby when I was living in the UK, and it was so interesting to see the role that cleanliness takes in modern parenting. That again, my fellow NCT mums, who are absolutely gorgeous and who I adore, and I'm still, very close to 10 years later, had such almost a fixation on the idea of bathing your baby every night. And I don't know what they thought of this feral Australian in their mix who did not bath her baby every night. But it was very much enmeshed in the idea of good parenting, that it's one of the things you do, at 6 o'clock in the afternoon or 5:15 or whatever your schedule is. I'm not a scheduled mum, by the way. whatever your schedule is, you get the bath running and you have your baby in the bath every night. And I remember kind of loosely trying to broach this and saying, look, this isn't necessarily the best thing for skin health and vitamin D, etc. But I learned pretty quickly that, you know, with your friends, it's always best to wait to be asked for advice rather than offer it unsolicited. But certainly this is something which feeds into the hygiene hypothesis, our keen interest or obsession with keeping our babies very clean. This is not the way that we build a healthy immune system. You know, we know that keeping children who, when we look at the epidemiology, children who are exposed to animals in the home, children who grow up on farms, children who are exposed to something that the researchers call farm dust, are far less likely to experience eczema. And, you know, we believe that this is due to their immune systems being exposed to a wider variety of microbes in the crucial formative time frame, which allows the immune system to learn to deviate, to distinguish friend and foe, essentially. So children need to be dirty, children don't need to be washed as frequently as we think they do. And there are many other things that feed into the hygiene hypothesis. It's not just dirt, it's microbial exposure. It's the fact that our children in modern life are less likely to be exposed to a range of childhood illnesses as previous generations. Obviously, this is a double-edged sword. We don't want our children to be exposed to life-threatening illnesses, but it does have immune consequences when they're not. We also know that the mode of birth plays a role in microbe exposure, something we've talked about many times, and the mode of feeding as well. And I have a little picture coming up to talk through this a little bit more. What's actually happening with the hygiene hypothesis on the immune system level when we have reduced exposure to microbial antigens? This decreases activation of toll-like receptors on dendritic cells and natural killer cells. So again, essentially, the developing, maturing immune system is not being presented with a wide enough array of microbial variety to learn about different pathogens and how to manage them. When we have reduced microbial exposure, which also correlates with a less diverse microbiome, this prevents that immune deviation from Th2 to Th1, which is this process which is controlled by the T regulatory cells. So we essentially end up, or we're more likely to end up with a Th2 dominant immune presentation, which drives the production of immunoglobulin E antibodies and the development of atopy. So essentially, if all the stars align, if you're more likely for this to happen if you have a genetic profile pointing you towards it. But remember, genes are not the whole story. If you've got a genetic profile, if you live in a less microbial diverse environment, if you have a narrower diet with fewer different foods, if you're born by a C-section, if you're not breastfed, if you don't go to nursery or other childhood daycare, if you don't live on a farm or live with animals in the house, you are less likely to have an immune system which appropriately deviates Th2 cells to Th1, and you are more likely to exhibit an imbalanced Th1, Th2 presentation. And this can certainly contribute to the development of atopy and allergy in general. What we see in patients with atopic dermatitis is levels of those Treg cells, which control the balance of Th1 and Th2, are decreased. And this, when we look at histological studies, this inversely correlates with symptom severity, the amount of circulating immunoglobulin E, and also eosinophilia. And those of you who spend a lot of time looking at blood tests, this is something that you know to look out for in your allergic and eczematous patients, is elevated eosinophils and elevated IgEs. So essentially, we are looking at an immune system which it's overreactive. You know, it is producing these IgEs and eosinophils in an attempt to protect itself from an enemy that doesn't really exist. This is one of my favorite pictures. Talk about this one for hours. I will try not to. This is representing what I like to call the diminishing human microbiome. And this is a hypothesis that I definitely am working with whenever we're working on new product development, or when we're looking at new research and how it fits in with clinical outcomes. We are always working with the assumed knowledge that with every generation in modern life, the human microbiome is becoming less diverse. Now, we've known for a long time that a more diverse microbiome correlates with greater health outcomes and is less associated with an allergic profile and an autoimmune profile. But there are so many different factors which feed into the diminishing microbiome. Like I said before, it starts with mode of birth, although to be honest, it starts way before that. It starts with your mother, and it starts with her mother, and it starts with her mother before that. For as far back as we can go, we know that the modern human microbiome on average is very, very sparse compared to what we call the ancestral human microbiome, that we have so many fewer varieties of microbes resident in our guts, on our skin, in all our body cavities, compared to 10 or 11 generations ago. And to be honest, compared with 1 or 2 generations ago too. So like I said, it starts with your mother and her mother, etc., because very roughly speaking, and this is a topic for an entire webinar on itself, but very roughly speaking, you can essentially only inherit your mother's microbiome. You can't really inherit, you can't really start life with a more diverse microbiome than your mum. So whatever microbiome your mum has, that's your top limit. And you will then spend the rest of your life essentially working towards that being your baseline microbiome. So how do you inherit your mother's microbiome? Both in utero, during birth, and probably more importantly, during breastfeeding as well. So the microbiome is, the fetal microbiome is seeded during pregnancy. And something that's really interesting is we know that women who take specific strains of probiotic while pregnant, those strains can be isolated from their baby's stool months later. So we do know that working on the maternal microbiome has a beneficial effect on the baby and likely their lifelong immune regulation. That's a pretty cool thought. Mode of delivery makes a difference. We are now looking at a world where in a developed nation such as Britain or Australia, between 30 and 50% of all births are delivered via cesarean section. Now, this is obviously crucial and life-saving for many women in many situations, but we also have to acknowledge the potential microbial consequences too. What's really interesting is that we're now gaining the understanding that most babies who are born via C-section, they will be at less of a microbiome disadvantage if they are breastfed. And breastfeeding appears to be more important in maintaining or building and maintaining microbiome diversity than mode of delivery. Now, this is something for you to keep in mind in Britain and Ireland, because Britain and Ireland together have the worst breastfeeding rates in the entire world. So it is no surprise that we see such a high presentation of atopic dermatitis in these societies, essentially. So what are the consequences of a less diverse microbiome? Essentially, in the context of atopic dermatitis, we are looking at a poorer innate immunity and a more reactive adaptive immunity. And this leads together to decreased tolerance, increased reactivity, and over the lifetime of this less microbiome diverse human, increased incidences of allergies and autoimmune diseases too. So we are essentially, by making our microbiomes less diverse, we are really setting the scene for immune dysregulation in the form of allergies and autoimmune diseases, depending on genetics and many, many other things. Right, what's going on with the skin? Let's talk about the importance of the epidermal barrier. The epidermal barrier is found in the lower layers of the stratum corneum. And this essentially is where we have highly differentiated keratinocytes coming together to form the barrier between the internal world and the external world. So if we think of skin as, basically think of yourself as a big sausage and think of your skin as the sausage casing. You know, it's holding all your insides together. and it's protecting you from the outside world. Well, the point at which the outside world stops and the inside world begins is the epidermal barrier. In where we see chronic and severe eczema, especially, we see significant damage to the epidermal barrier. And there are many factors, once again, that feed into this damage. From the external world, anything you put on your skin has the potential to contribute to epidermal barrier dysfunction. So this includes, well, this is an interesting conversation, because this includes not just things like steroids, which ironically are used to treat eczema, more on that later, and personal care products, but something that is highly relevant for all of us in the world at the moment. There's a significant relationship between use of antibacterial personal care products and disrupted epidermal barrier dysfunction. So think about what we've all been slathering over ourselves and our children for the last year or so. You know, again, there are reasons for it, but in the context of epidermal health, this hasn't been a great thing. Steroids are interesting because steroids are obviously the first line treatment for eczema. And the reason for that is because they are significantly anti-inflammatory and immunosuppressive. So they will reduce the immune reactivity present in and around the epidermal barrier and reduce the symptoms. However, long-term use further weakens the epidermal barrier, and I'll show you on a picture coming up, can feed into that cycle of barrier dysregulation. Now, I'm not a practitioner who's ever discouraged anyone from using steroids when they need them. Like that case I was describing to you earlier, that woman, bless her heart, she had tried so hard not to use steroids on her hands, and she was in such a mess. I said to her, look, just put your steroid cream on right now, and we are going to work from the inside. But while you are in such a state of inflammation and pain, we're not going to get anywhere. So steroids absolutely have a role to play. But what we want to do is help our patients move away from the cycle of steroid dependence, where they are continually feeding into epidermal barrier. It's too early in the morning to say this word, epidermal barrier dysfunction, which is contributing to keeping them in this vicious cycle. Something else that's really interesting is stress is a trigger. And those of you who have atopic dermatitis yourself, you know, I am very sure you will agree with this, that in adults especially, where the incidence is lower, I remember so many of my patients and my students saying to me, you know, I haven't had eczema since I was a kid, but in the lead up to my wedding or, you know, final exam or divorce or whatever, my eczema has broken out again. So it's very well recognized that psychological stress is a trigger, and it's also a consequence of atopic dermatitis too. Stress hormones can inhibit the synthesis of some of the lipids needed to maintain the hydrophobic skin barrier and further feed into that epidermal barrier dysfunction. What we're looking at when we start to see dysfunction is transepidermal water loss. That's what that TEWL stands for over there on the left, transepidermal water loss. This is so interesting because essentially the stratum corneum contains something we call the NMF, or the natural moisturizing factor. And the natural moisturizing factor is essentially a collection of filaggrin metabolites which hold moisture in the stratum corneum. Now I have a picture of this coming up, just kind of introducing this, and I just love talking about this because, you know, I love a good bit of pathophys to start me off in the morning, and I love understanding, you know, what's going on. So when we come to talk about filaggrin, we'll go pretty hardcore on the natural moisturizing factor. So we have transepidermal water loss, dehydration, inflammation, dysfunctional keratinocytes, which then, remember, it's highly differentiated keratinocytes which build the stratum corneum. When we have dysfunctional keratinocytes, they're not able to as effectively maintain the stratum corneum, and therefore epidermal barrier dysfunction occurs. What's interesting though is when we have this water loss and the dysfunctional keratinocytes, this itself leads to increased Th2 production as well, or reduced deviation from Th1. So the epidermal barrier, this is where we start to see that vicious cycle building up. We've got immune dysregulation, which causes a breakdown in epidermal barrier function, but then the more damaged the epidermal barrier is, the more dysregulated the immune system becomes, and round and round and round we go. This then elevates the risk of infections, specifically Staphylococcus aureus. And Staphylococcus aureus, it turns out, is the big baddie when it comes to worsening atopic dermatitis for several reasons. And I've got some pretty cool slides coming up a little bit later. So what we see clinically is dry skin, because we've got this transepidermal water loss and a reduction in natural moisturizing factor. We see inflammation because we're stuck in a cycle of locally produced cytokines, especially interleukin-4, interleukin-13, both of which drive elevation of Th2. And then when we have elevated Th2, this drives further immune dysregulation, leading to itching, scaling, swelling, edema, infection risk, and round and round and round we go. It sounds like a lot of fun. So I mentioned filaggrin a second ago. Filaggrin is a protein found in the epidermal barrier, well, found in many layers of our skin, actually. It's a really important protein for maintaining that natural moisturizing factor. Now, there are a couple of ways of looking at filaggrin in the context of atopic dermatitis. First of all, there's a particular genetic polymorphism which is linked with a reduced ability to produce filaggrin. And so this means this is one of the factors that leads into the genetic aspect of eczema. So it's an inherited disposition to produce less filaggrin. But if we look at the statistics, remember, genetics are not everything. Only about half of all eczema patients have this genetic polymorphism. So there is obviously, many other factors at play. It's not just genetics. And also that means between 40 and 50% of people who have the gene do not express eczema. So it's never that simple. It's never black and white. What we see more, I don't know if it's more common, but what we see in the remaining the remaining half of all eczema patients who don't have the gene, and also plenty of them who do, is acquired filaggrin deficiency via Th2 dominance. So remember that Th2 dominance cycle that our patients are trapped in. This dominance of Th2 further suppresses the expression of the filaggrin gene. So essentially, the more inflammation, the more immune dysregulation, the less able you are to produce more filaggrin. So what's important about filaggrin? Filaggrin is what we call an upstream protein for maintaining the natural moisturizing factor. When filaggrin is metabolized in the stratum corneum, it produces 4 specific metabolic proteins, or metabolites, I guess: carboxylic acid, urea, lactic acid, and urocanic acid. And these 4 metabolites, I'll just repeat them again: carboxylic acid, urea, lactic acid, and urocanic acid. These 4 post-metabolite proteins together create an osmotic draw in the epidermal barrier. So the presence of these proteins in the stratum corneum draws moisture from tissues to be held in the epidermis. And this is what we refer to as the natural moisturizing factor, or the NMF. And this is what helps keep skin essentially moisturized from the inside. When we have a deficiency of filaggrin, either genetic or acquired, we are less able to produce these downstream metabolites, and therefore less able to hold moisture in the epidermis and the epidermal barrier. This then kicks us off into that cycle of dryness, itchiness, inflammation, damage, etc. So filaggrin, it turns out, is one of the big moving parts in the incredibly complex machine of maintaining skin health. By the way, think about all of those metabolites: carboxylic acid, urea, urocanic acid, lactic acid. What do they all have in common? They're all acidic. So they help together to maintain the skin pH and the ideal pH of the skin, and to keep that pH low, essentially. When we see an increase in pH of the skin, this is when we invite microbiome dysfunction on the skin surface. And this is when we're more likely to see colonization with Staph aureus again. So maintaining skin pH is a super, super important function of natural moisturizing factor and therefore filaggrin. But remember, filaggrin's only one piece of the puzzle. About 40% of people who carry the gene for filaggrin deficiency, they don't develop eczema. So it's not, it's not black and white. It's not the be all and end all. We've always got to look at all of the factors going on. My favorite slide in the whole presentation. This is so cool. So let's talk about the bidirectional relationship of skin barrier dysfunction and Th2 immune cell dysregulation, or dominance, really. So remember, we've got this vicious cycle going on where we have barrier dysfunction leads to elevated Th2 leads to barrier dysfunction. Round and round and round we go. Let's have a look at what's going on in this picture. These are those differentiated keratinocytes, which we know as corneocytes, okay? It's the corneocytes which are found deep in the stratum corneum. They are the beige rectangles there. Now, the little purple spheres that you see at the ends of each beige rectangle, these are the corneodesmosomes, and these are little proteins which hold those corneocytes together. When we see damage to those corneodesmosome proteins, this is when we see a breakdown of that epidermal barrier. The little P's we have floating around here are proteases. So what are proteases? They're enzymes which break down proteins. What are these little corneodesmosomes? They're proteins. So when we have elevated presence of proteases, we have a breakdown of the cells which are holding the corneosomes together. Now, where are these proteases coming from? Lots of different directions, actually. In this picture, we see both exogenous and endogenous proteases. Exogenously, so coming from the outside world, proteases can be coming from metabolism of Staph aureus. So if we have an overlying Staphylococcus infection, as this bacteria embeds itself on the epidermis, it produces proteases, which further damage the epidermal barrier. Dust mites, that's what that's big, scary, kind of hairy thing is over on the right there. That's a dust mite. And again, I'm sure that we're all aware of the relationship between dust mite allergy and the atopic presentation. Dust mites, it turns out, also produce proteases. So when we have a dust mite heavy environment, these little critters crawling around us in our sheets, etc., they are also producing proteases, which are further delivering that skin barrier breakdown. But we're also producing proteases endogenously in a Th2 dominant situation. So we've got our immune system going on down here. This is a dendritic cell, and here it is presenting an allergen. Now, this allergen has likely come from, or has come from, the external environment. And this is, again, another really interesting topic, looking at how exposure to environmental allergens via epidermal barrier dysfunction increases sensitization to those allergens and enhances the allergic march. So essentially, if you have reduced barrier dysfunction, more allergens are allowed into your skin, where they are then kind of picked up by dendritic cells and presented to your Th1 cells. And this is why when we have people with broken skin or very damaged barriers, we have to be so careful about what we put on that skin. And this, it's interesting, researchers have found a strong correlation between using food-derived ingredients in eczema creams and allergies to those foods. So we need to be careful about what creams we're actually putting on the surface of children, especially children with severe eczema, and try to keep them as non-allergenic as possible. Unfortunately, this does rule out a lot of the kind of natural creams and things that we love to use, but that's a whole different presentation. What I want to point out is that allergens making their way through a damaged epidermal barrier can promote the march of allergies and actually exacerbate the allergic response to external allergens. So here we have a CD4 T cell here. which is enhancing the production of interleukin-4. So it's been recruited by the presence of elevated allergens. And Th2 also recruits CD4 cells to the local environment. So when we have, so we've got more allergens which are being presented to Th1, which is then hastening the deviation of Th1 cells to Th2. When we have higher levels of Th2, the Th2 presence also recruits CD4 cells to the area. The more CD4 cells we have, the more we are triggering production of interleukin-4, which itself also increases the deviation from Th1 to Th2. Th2 enhances production of interleukin-13, which further dysregulates the stratum corneum breakdown. So we're stuck in this vicious cycle where every part of this picture is feeding into every other part. And it can be so difficult to break this vicious cycle. Here's a picture for you to all print out and study if you would like to. It's fascinating, and my colleague Ben, who put this together, just had way too much fun doing this. He was absolutely blown away when he was reviewing all the research at the way that every part, like I just said, every part of all of the factors that drive atopic dermatitis feeds into every other part. Just briefly, because I don't want to take up too much time on this when I know that you can, you can all print this out and read it in bed. If we start here at elevated Th2, I mean, you can start anywhere in the flow chart. That's the beauty of it. But if you start here, we can see that when we have an elevated Th2 presentation, this is reducing the production of filaggrin. When we reduce the production of filaggrin, this is one of the things that feeds into skin barrier dysfunction. But remember that elevated Th2 increases that epidermal barrier dysfunction, dysfunctional skin cells. We're more likely to see Staph aureus colonization. Staph aureus itself increases the presence of Th2 cells. So this is one vicious cycle going round and round itself. When we have Th2 immune cell dominance, we have elevated interleukin-4, interleukin-5, interleukin-13, all of which increase physical inflammation and the likelihood of allergies. And these also drive the production of immunoglobulin E antibodies. So we are essentially making someone more allergic when we have this Th2 immune dominance. What's feeding into the Th2 immune dominance? Well, the dysbiotic environment we talked about, the diminishing human microbiome, altered immune system maturation, and on we go. When we have these elevated IgE, we're more sensitive to allergens, but we're also more sensitive to allergens due to the skin barrier dysfunction over here, which may be contributed to via filaggrin mutations, stress, environmental factors. Then we're stuck in that Th2 cycle, feeding further filaggrin dysregulation. We have infiltration of allergens into the skin, which is then feeding the allergic profile, sensitivity to environmental irritants, exposure to chemical irritants, creating more inflammation, leading to more scratching, which creates more dysfunction and more inflammation, more opportunities for Staph aureus colonization. So then we're feeding back in here as well. And it's all adding up to an atopic dermatitis presentation, which, if we're really lucky, insert irony, we then move further along the atopic march. And we know that atopic dermatitis is just the beginning of the atopic presentation. Children who develop eczema are more likely to develop allergic rhinitis, asthma, and food allergies too. And, I'm very firmly of the belief that it all starts essentially with atopic dermatitis. So that is a brief introduction to all of the factors playing into atopic dermatitis. What can we do about this? We've already mentioned the problematic nature of using topical steroids. This may interrupt that itching discomfort cycle, but it's unfortunately only going to be contributing to further disruption and maintaining all of those vicious cycles. We really have to look at what's going on the inside. As we've acknowledged that microbiome dysregulation is one of the factors leading into this vicious circle, let's look at it as one of the factors leading us out of that vicious circle. So we've had a look at the research around specific strains of probiotics and what they can do in this situation. And there's one strain that stands out above all others, and that is Lactobacillus salivarius LS01. So those of you who have seen me talk about probiotics before, you'll know that one of the kind of points of difference, or one of the things we're most passionate about at Activated Probiotics, is always talking about the specific strain of probiotic that we use and all the research on that specific strain. So when we're talking about probiotics, it's really important to look at the full name, the genus, Lactobacillus, the species, Salivarius, and the strain. And in this case, it's LS01. It's this research that we're about to talk about is absolutely not pertinent to any other Salivarius strain, just LS01. And in fact, LS01 has been trialed against other Salivarius strains and found to be significantly more effective. So it's not enough to just look for a Salivarius, it must be Salivarius LS01. Of all the strains we use at Activated Probiotics, this one is in my top 3. It's either number 2 or number 3, depending on which product I'm working with most. I really, really like this strain. And I think that its potential in helping to stop that allergic march is huge. Very, very keen on Salivarius LS01. Let's have a look at it in the context specifically of atopic dermatitis. Now, like I said, one of the reasons I like this strain so much is that it has a really decent amount of published research available on it. And again, those of you who know either me as a lecturer or Activated Probiotics as a company, you know that we are very keen on clinical evidence. And this is, again, one of the reasons I like this strain so much, that it has been looked at from lots of different angles in published trials on various factors of atopic dermatitis. Now, LS01 has actually been found to be able to reduce the SCORAD, which I'll show you in a second. Essentially, it's a marker of severity, or a tool used to measure severity of eczema, by 52%, which is absolutely not insignificant. What we see in this particular study is after 4 months of administration of LS01, we have that hugely significant reduction in SCORAD score only in the LS01 group, or no changes reported in the placebo group. Now, the first thing I want to point out is that whenever we are working with skin conditions and the microbiome, this is not an overnight fix, okay? Think about all of those factors feeding into the epidermal barrier dysfunction. This is not a vicious cycle that can be turned off overnight. We need to look at a full skin cycle under the influence of lowered inflammatory conditions. So generally, the time frame that we're working on to see improvements in eczema when using probiotics is at least a month, more likely in the 2nd month, where we see benefits. So this is something I always talk to my patients about, to say, look, we are going to see improvement for you. You know, I'm very confident of that, but this is a condition you've been living with for a long time. You're in a severe cycle of immune dysregulation and inflammation. This is not a quick fix, but it is, you know, there's something that we can absolutely do to help regulate your immune system and make this less likely to happen, and to happen less severely for the rest of your life. But we generally are looking at the 2nd month of treatment being the most relevant month. So what is SCORAD? SCORAD, like I said, is an internationally recognized tool that's used to assess the severity of dermatitis. So it stands for Scoring Atopic Dermatitis, very creative there. And it's a standardized tool. It's measuring lots of different things. So it's not just measuring, you know, the percentage of the skin affected or the redness. It's looking at the redness and the swelling and assessing the presence of any oozing or crusting, scratch marks, skin thickening, dryness, all of the physical manifestations of dermatitis. It also asks patients about their sensation of itchiness and the sleep disruption that they experience too. So it's a pretty holistic tool used to assess the quality of the severity of eczema. And it results in a score, essentially. So what we see when trialing LS01 in eczema patients is a 52% reduction in that score. That's huge, huge. We also see a 55% improvement in quality of life. And, you know, remember I said earlier, this makes my heart sing as a naturopathic professional, because we're really looking at the holistic aspect of our patients' lives. We're not just looking at, well, you've got less redness, so you're cured. We're looking at how is this affecting every aspect of your life. There's a tool used to assess this in eczema patients called the Dermatology Life Quality. DLQ, which is a PROM or a medical questionnaire. And again, what we see is a hugely significant improvement in quality of life in eczema patients in those who take LSO1 compared to placebo. Again, let me point out the time frame. This significant effect was observed after 2 months. Okay, so once again, we always need to talk to our patients about time expectations. You know, we're not here for a quick time, we're here for a good time. Okay, we're looking at the long-term fix. We're not suppressing the symptoms, we're not turning anything off. We are working with the body to break those vicious cycles and help to normalize the immune response. Remember that what we're looking at is always a complex interplay. So if we have a think about our mechanisms of actions of LSO1, what's it actually doing? We know from histological studies that it is modulating that all-important Th1, Th2 balance. So remember, so much of what we've talked about tonight has been the problem with elevated Th2. If we can help normalize and create a more favorable Th1, Th2 balance, we can really cut into that vicious cycle of Th2 stimulating CD4 recruitment, interleukin 4, interleukin 13, proteases, skin breakdown, etc., etc. So that's huge. And also think about the implications that has for us as naturopathic practitioners for other Th1, Th2 dysregulated patients as well. LSO1 can also help with repairing gut integrity and reducing LPS translocation. Some information on that coming up in a second. And significantly inhibit Staphylococcus aureus too. Now we've introduced Staphylococcus aureus as the villain of tonight. Let's talk a little bit more about why it's such a baddie. Do you like our balancing rock picture? Our digital marketing manager was trying to find a picture to represent Th1, Th2 balance, and he came up with rocks. There you go. They look very pretty though. So when we talk about Th1, Th2 balance, when we help to improve this ratio, we are reducing allergen-specific IgE antibody production. That's huge. Do we understand the implications of that? We are essentially making someone less allergic when we give them Lactobacillus salivarius LSO1. You know, this, to me, this is one of the most exciting things I've ever worked with in the world of probiotics, that we have this all-powerful strain which can help make an allergic person's immune system behave better and become less responsive to allergens, essentially become less allergic. Huge. When we look at that Th2 overpresentation and the effect it has on filaggrin deficiency, we are helping to promote improved skin barrier function. We are promoting the production of antimicrobial substances in the skin, and this is another factor that helps to protect against Staph aureus colonization, along with helping to maintain correct skin pH and that natural moisturizing factor that holds water in the skin. And we're also obviously reducing symptom severity for all of the reasons that Th2 drives inflammation and itching. Some really interesting blood studies done on different strains of probiotics found that the Salivarius LSO1 strain in an allergic population really modulated that balance and favored a Th1 response, whereas other strains of probiotics actually helped to drive a more pro-inflammatory Th2 cytokine response. It's just another indication that we must always look for the specific strain. Remember, it's not enough to be species specific, we need to be strain specific. It's not all Salivarius that does this, it's specifically LSO1. So another really interesting aspect of LSO1, and when we look at these kind of histology pictures, is its effect in allergic rhinitis. And this won't be any surprise because, as we've already acknowledged, patients who have eczema go on to develop hay fever. So LSO1, this is now my new favorite strain for the hay fever season, which I know you're just coming into over there, because I remember every May, my poor allergic husband would dissolve into a snotty, sneezy, grumpy mess. And, you know, out would come the annual packets of antihistamines that he kind of, you know, had to live on for a few weeks. Well, let me tell you that when we moved to Melbourne, Australia, oh my goodness, that was ramped up by about 100-fold For various complicated geographical reasons, Melbourne is the hay fever capital of the entire world. And it is so common to meet people who have moved here who say, look, I never had hay fever before until I moved to Melbourne. So his hay fever went out of control the first year we were here. It wasn't like in the UK, it would last maybe 4 weeks. Here it lasted 5 months. And he was the first 2 years we were here, he was taking a daily antihistamine for 5 months straight, sometimes 2 a day. That was before we discovered LSO1. So last summer, I had him on our Biome Breeze probiotic. I gave him 2 sachets a day because he was, honestly, talk about quality of life. My quality of life was being ruined living with him. I can say that because he's still asleep in bed. And it just stopped. It stopped. And at about 3 months into the season, he kind of said, I've only taken 2 antihistamine tablets this entire season, whereas he had been taking 2 a day. So I am such a huge fan of the effect of LSO1 on allergic rhinitis, and it's so great to look at what it's actually doing. So it is really working on peripheral mononuclear blood cells and helping to downregulate those cytokines, which again recruit Th2 involvement and promote the production of IgE, specific IgEs, and really contribute to that allergic march. So what we saw in the studies comparing LSO1 to placebo was that while the LSO1 group, their cytokine levels remained stable in the face of allergic rhinitis season while taking LSO1. The placebo group, it demonstrated exactly what you would expect, which is increased interleukin-4, decreased interferon, and increased Th2. And what we recognize from this is that LSO1 can induce those all-important Treg cells, which monitor or adjust the deviation of Th1 to Th2 and really help to improve tolerogenicity and that Th1-Th2 balance. So I promised you earlier some chat on intestinal permeability. I'm very aware that I'm talking way too much, so I'll just try and kind of fly through the rest of this to leave time for questions as well. Although anyone who has to go, you can always watch the rest on replay. Gut repair. We always talk about gut repair in the naturopathic world, and that's what we use probiotics for, right? Well, in this case, what we're using probiotics for is Th1, Th2 regulation and epidermal barrier function, etc. But yes, we are also working on gut repair. When we see allergic people, we are very, eczematous people, we very often tell them, look, your skin is like a reflection of what's happening in your gut. Just as we have leaky skin, it's another way of thinking about that epidermal barrier dysfunction, we very often see leaky gut as well. And this can be driven by LPS levels, lipopolysaccharide endotoxins, which are coming from gram-negative bacteria in our digestive tracts. And they can negatively affect the tight junctions between enterocytes and lead to what we talk about as increased intestinal permeability or leaky gut. Translocation of LPS, if they can actually sneak through that hyperpermeability situation into the peripheral blood stream, they will then trigger further immune activation and inflammation and therefore exacerbate symptoms of atopic dermatitis. We do know that that elevated Th2 presentation positively correlates with plasma LPS levels. So once again, if we can reduce gut permeability, we can reduce that march of Th2 and everything that it feeds into. Just a quick comment on this study on LSO1 and LPS concentrations in the blood. Patients who took in the trial LSO1 compared to placebo had significantly less microbial translocation during treatment, and how exciting is this, 2 months after treatment finished. So this is what I love about probiotics. Remember, a great way of thinking about them is they're like getting the builders into your house. They come in, they do renovations, and they leave the house in a better state than when they arrived. So whenever you're using probiotics, you are working with the body to bring itself back into balance. So these people still experience great benefits 2 months after they'd even finished taking LSO1, okay, which is a brilliant thing to think about in your treatment protocols. So by reducing serum levels of LPS, LSO1 showed that it has the ability to help essentially gut healing and reduce the antigenic load on the immune system. Now, I said before that Staph aureus is the big baddie in this situation. Several reasons for this, but one is that it's highly likely to present as a comorbidity in severe eczema, and this is the infection which is being treated when patients with eczema are given antibiotics, essentially. The reason Staph aureus is such a baddie is because it can possess what we call super antigenic exotoxins. More on that in a second, and can kickstart or further feed into that Th2 recruitment, inflammation, etc. Now, something that's super interesting is, we were saying earlier about 20% of kids have eczema, but only 5% of adults. Why is that? Why is eczema something that we grow out of? Turns out that one of the factors may be the changes in microbiome that occur as we move from a more juvenile microbiome to a more mature microbiome, because an adult microbiome has a higher prevalence of bacterium that produce specific anti-staphylococcal peptides, and children don't. So essentially, we become better at protecting ourselves, I guess, against Staphylococcus infection and proliferation as our microbiome shifts and matures. So this again comes back to the idea of looking at the microbiome as a really important place to start in treating eczema in the first place. So what are these super antigens? Super antigens produce 10,000 times greater lymphocytic recruitment than normal antigens. So if we think again of those Th1, Th2 lymphocytes, when we have super antigens present on the surface of the skin and infiltrating the immune system due to that damaged barrier function, we are actually further significantly recruiting Th2 deviation and CD4 involvement. This can also lead to greater production of IgE. So essentially, the short way of talking about Staphylococcus aureus is that it is super inflammatory and super immune dysregulatory. So it is contributing to that Th2 dominance significantly. And this can significantly worsen skin inflammation and persistent sensitization to allergens, and is probably key in the development of food allergies. Okay, but that is again a topic for another day. So what have we got happening with LSO1? We see in the context of Staphylococcus, in double-blind placebo-controlled trials again, we see a significant reduction in the number of staphylococci that are actually able to be colonized from stool samples in people taking LSO1. And that is after taking the LSO1 for 4 months, but also after a 1-month washout period too. So the LSO1 has a continuing ongoing protective effect against Staphylococcus aureus colonization too. So where do we get this magic goodness of LSO1? We have this strain in our Biome Breathe formulation. And this is interesting because Biome Breathe is called such because it has been specifically trialed in the context of asthma. But what is asthma? It's a continuation of the allergic march. It is no surprise to us that the same people who present with asthma previously presented with atopic dermatitis. They are essentially different manifestations of the same dysregulation, the same Th2 dominance, the same IgE overproduction, the same elevated levels of interleukin 4, interleukin 5, interleukin 13. So it is no surprise at all that Biome Breathe is also so brilliant for both eczema and atopic and allergic rhinitis too. So Biome Breathe contains Salivarius LSO1, as well as a really fascinating bacterium we'll talk about another day, Breve B632. We are also, those of you who have tried it, you know that it comes in these individual sachets like this. It is an unflavored powder. It's completely inoffensive. You can mix it in water and it is totally flavorless, colorless, odorless, etc. But those of you who have tried our kids product and you know how delicious that is, we've had so much feedback on how much people love our kids probiotic that we're actually reformulating the flavor of Biome Breathe in the future. So from later this year, Biome Breathe will also be that lovely, delicious vanilla flavor that we have in the kids product, which is free from sugars and artificial sweeteners and melts in your mouth. So it will be, I guess, more of a flavor treat for kids you are wanting to treat for eczema. We also have in the future, we've been working on different formulations, etc. We also have an eczema specific product coming out very soon in the next couple of months, should be hitting the UK and Ireland. In the UK and Ireland, it will be called Biome Soothe. In Australia, it's called Biome Eczema, but your regulatory authorities won't allow us to call it that over in your part of the world. So we've called it Biome Soothe, and it will contain just Salivarius LSO1, no Breve. And it will also be in the flavored fast melts. So Biome Soothe is coming. However, in the meantime, we have Biome Breathe, which contains that delicious Salivarius LSO1 strain that I love so much, and will be, available for you, well, right now, just in time to get all your allergic rhinitis patients on as well. So that brings me to the end of my too long waffle. I know that lots of you may have to go, but I'll have a look at the questions, and let's chat through what your questions are.
Okay, Beck. So the first question is, wondered if this would also help with rosacea.
Oh, interesting. If it is an allergic mediated rosacea presentation, absolutely. And especially if there is a food allergy component, because, like I said, what we see is very much that Th2 dominance being involved in a food allergic picture. So if there is a food allergy component to the rosacea, then yes, absolutely. Something else that I didn't mention about the LSO1 actually that I think is probably really relevant to many of you is that this is also the strain we recommend for histamine sensitivity, that it is a mast cell regulating strain as well, which is no surprise given that it's a potent anti-inflammatory and anti-allergic strain. So this is also the strain that we recommend for those really histamine sensitive patients to help get their immune systems back on board with histamine regulation. Sorry, Karen, go ahead.
What's the name of the gene affecting filaggrin?
Oh, I don't know. It's in the reference that we've provided, so you can look it up.
I thought you might know off the top of your head. That's all right. That's fine.
My powers don't extend to that at 5 o'clock in the morning, I'm afraid.
Just to let everybody know that I can send the slides out to you, just email Karen at activated.co and I'd be more than happy to do that for you all. Does any of this apply to psoriasis, or is it another matter altogether?
Yeah. So psoriasis is, even though it is another skin presentation condition, we think of it in a totally different way, because psoriasis is, I never call it a skin condition, I call it an immune condition. So the strains we would look at for psoriasis would be coming in from a different angle. We're not necessarily looking at such an allergic profile with psoriasis. We're looking at an immunodysmodulation profile. And I'm going to be looking at that with a few other strains in mind. Some of the strains we have in our Biome Advanced product, or this is going to sound like a strange thing to say, but I'll cover this in a future webinar. Our Biome Osteo product is actually what I would look at for psoriasis. Having said that, when you've got psoriasis with Staphylococcus aureus infection and epidermal barrier dysfunction, then yes, the LSO1 can be helpful there too.
Okay, thank you. I think we've got the Osteo one later in the year actually, so that'll be something to look forward to. Right, okay, so yeah, recordings, no problem. Right, well, this is Helen. She's getting a bit personal now, bless her, but we love Helen, so that's okay. What level of allergy would you use to help you to decide that your hubby or anybody needed 2 capsules per day or 2 sachets per day? Where is she on the scale? I think that's what she's trying to work out.
That's a really good question. Generally, all of our products are based on, we've formulated them based on the research to give 30 doses in each box. And we generally say 1 dose a day. However, he was honestly just such a grumpy pain in the bum that I'm like, right, I need to get this under control so quickly. So I'm going to give you 2 strains a day. With probiotics, and this is probably again a discussion for a longer webinar, with probiotics, it's not always the case that a higher dose means more significant effect. So it's not really as simple as you increase the number of billion live bacteria to enhance the strength of the effect. but it can, in some cases, elevating the number of live bacteria can speed up the immune response. So if you've got a patient with really severe eczema who's having a particular flare, then yes, I would use 2 sachets a day, especially in adults. For children, I'd probably stick with 1 a day. But you're still going to get to the same place, okay? You just might get there a little bit faster if you use 2 a day.
Mm-hmm. Okay. And we need to do that if you've got a grumpy husband. So that's
okay. Oh yeah, it was a cure. It was a marriage saver, I tell you.
So the next question is, would this help with people with Lyme's?
Oh, that's a really good question. I haven't seen any research on that specifically, so I don't feel confident, but have a look at the histological profile of your Lyme patient. If you are looking at a situation of elevated Th2, elevated interleukin-4, elevated interleukin-13, then I'm going to say probably yes, but it will depend on the level of immune dysregulation and which pathways are being upset.
Okay. When would you start treatment for hay fever? Either 1 to 2 months before the season, or when the hay fever actually kicks off?
Well, there are 2 ways of looking at that. One is what should you do? And the other is what actually happens in practice. So obviously we all know that prevention is better than cure. And ideally, I would say, look, start Biome Breathe a good 2 to 4 weeks before you think you're going to need it. But what happens in your clinical practice? Patients turn up with hay fever. So in that case, you just get them on it ASAP.
Fabulous. Is Biome Breathe suitable for children? I think
yes. And it actually, when we'll have, I know we've got another webinar coming up at some point to talk about the trial that was done on Biome Breathe in the context of asthma, particularly. That trial was done in children specifically. So yes, it absolutely is. Just what's interesting is that we have to change our wording on the packaging in different regions. I think in the UK and Ireland, the wording on the packaging is for children 3 years and up, or under direction of health professional. That's not anything to do with the safety profile. That's all to do with regulation. We don't have that wording on our Australian boxes. Okay. So the trial did include children younger than 3, and there are absolutely no safety concerns that we are aware of using it with younger children. Okay. Just to let you know that it appears on the box, not from a safety angle, but from a regulatory angle.
And if anything, it's better for children because it's already in the powder form, isn't it? You just take it out of the sachet and put it into something if that's what you want to do.
And they honestly, it's completely flavorless. They'll never know it's there.
So it's easy to hide. Hide in those vegetables that you're trying to hide from them. There you go.
So parenting is just a long journey of deception, isn't
it? Right. Okay. Interesting. Are these products available in the US?
We don't have any wholesale distributors in the US. The US is an entirely different market for so many reasons, but American patients can contact us via our Australian website and talk to us about that.
Lovely.
And of course, if we have US patients, we can mail them to them. Yeah. Sorry, Karen.
Okay. Will LSO1 be good for reactive hives?
Yes, because what we have in reactive hives is a massive histamine release, and LSO1 is a histamine stabilizing strain, as well as helping to reduce IgE levels and the various interleukins that lead to histamine response. So yes.
Mm-hmm. Okay. Would it be suitable for patients with stomas, etc., and Crohn's disease, or anybody that's got any form of damage, sort of gut or IBD, as it says here?
Yep. Short answer is yes, it is suitable because of its ability to reduce LPS translocation. The longer answer is there may be other strains more relevant. For IBD patients, we generally look at either a Biome Advanced or Biome IBS. Sorry, that's called Biome Relief in the UK. So either Biome Advanced or Biome Relief. But to answer the question, yes, it's absolutely fine.
Yeah. Okay. Is it safe with blood thinning medication?
of my favorite questions, because the answer is yes. And again, that's why I love working with probiotics so much, because they have such a great safety profile. They're not contraindicated with any medications.
Is it vegan?
Oh, yes, it is. So, yep, another favorite question. So we are very much a plant-based company, and all of our probiotics are dairy-free. They're all egg-free. They are all vegan, with the exception of 2, which use lanolin-derived vitamin D. That is Biome Osteo and Biome Prenatal. But all of our other probiotics are vegan, yes.
Okay. Is LSO1 affected for eosinophilic osteophagus? I hope
Yes,
I said that right,
yeah.
which is in response to a milk protein allergy.
Yes. I know exactly what you're talking about, eosinophilic esophagitis. And yes, it is, because what we see in eosinophilic esophagitis is dramatically increased production of eosinophils, which is, yes, usually in response to milk protein allergy. And this is an absolutely brilliant application for LSO1 for sure, because what we're doing is interrupting that inflammatory cascade before it even gets to the point of triggering eosinophils, well, hopefully. So what we see in eosinophilia is, significantly elevated IgE and Th2. And as LSO1 disrupts the pathways that feed into production of both of those immune products, yes, it absolutely is. I'm getting very emphatic about this, because I've seen so many babies really nastily affected by eosinophilic esophagitis. And I'm so, I just feel so pleased that we have something safe to offer them as part of the treatment profile.
Okay, so this is a good one. If a patient experiences mood problems, but as well as skin problems, would you recommend using 2 different of the probiotics, i.e. would you use Biome Lift and Biome Breathe?
Well, you can. There's no problem with using those 2 products together. However, you may find you don't need to. And it's such interesting feedback we get from so many of our patients and clinicians is that very often they are kind of choosing between, they say, I've got a patient with a particular presentation that fits one of your products, whether that's iron or IBS or osteo, or in this case, breathe. But my patient also has low mood and poor sleep. Should I use both? And my answer is always, you can use both, but let's just try using the condition-specific product for a month first and see what happens. And so often they will come back and say, well, my patient is sleeping better, she's feeling better, her mood is improved through using Breathe or Osteo or Advanced. So, to sum up my answer, yes, you can use them together, but you may not need to. I would go with a month of Breathe, or maybe 2 months, and see.
Fab. Okay. So this question says, would this be regulatory enough in the case of autoimmune thyrotoxicosis, or would that mirror the psoriasis and be more advanced than osteo? Great
question. I'm going to suspect would be looking more at the immunomodulatory strains. So that would be the Plantarum Heon9, Paracasei 8702. So I'd probably go for osteo or advanced in that situation. But having said that, I'm just thinking of some feedback we had last summer from a gentleman who was using Biome Breathe for allergic rhinitis and found significant improvement in his autoimmune disease as well. So, let's face it, probiotics just fix the whole world.
Absolutely. I think you sort of answered this. Sophie's saying, can you mix and match from the activated probiotics range and use them together?
Yeah.
Kind of
covers a little bit. Yeah.
Yeah, okay, that's fine. Right. Can you give these probiotics to somebody with a corn allergy?
Theoretically, yes, because there shouldn't be any corn protein present in the finished product. There's no problem with corn allergy. I've just spotted another interesting question actually in the question channel. Just opened it up. What's your opinion of the biologic agent dupilumab? And that's I'm just going to highlight that for a second, because I was actually just in a meeting with a pharmacist yesterday who was talking about eczema in particular, and that particular medication, and asking about how Biome Breathe would complement it. And it turns out it does complement it beautifully, because this drug essentially is an immunomodulant which results in reduced levels of Th2. So this is also ultimately what we're aiming for with Biome Breathe. So my opinion of that drug is, I think for some patients it has been helpful for them, but it's a pretty hard hitter, and I would love to see patients trying Salivarius LSO1, preferably before they get to needing, you know, a Th2 regulating drug. Cool. I reckon we're probably extended the limits of everyone's patience today. So I might let you go, but if any other questions that pop up, you can always email Karen, or get in touch with us, or Josh, get in touch with us on our Facebook group or our website. Josh, did you want to add something?
I did, very non-technically, in that exactly what you were describing through that. I was exactly that child and wished that had come along with the milk allergy, and still a little bit of eczema and hay fever particularly, and it's working for me. So I've been on that a month. The eczema's gone from my hands. I won't show people my grotty hands, and I've given it to my brother, who's now started it as well, who's the same, because it's obviously a genetic thing. He gets it terribly and is battling away, and he's been on it for a couple of weeks. As you said, should have done it weeks and weeks ago, but of course you forget until you're suffering. And it's doing the business for us. So
I'm very pleased. Fantastic. Wonderful. I'll let you all get on with your evenings. I'm going to go and pack school lunch boxes. So I will look forward to seeing you all next time. Come and say hi in our Facebook group, which and you know, once again, I look forward to seeing you all in person when the world allows it. Come over and visit. But in the meantime, I love seeing you all on screen. Hey, see you everyone.
Thanks.